Oh, okay. Hi, everybody. Welcome to the NCDS DataBytes Lunchtime Webinar Series. Today, our speaker is Stephen Edwards. He's a Systems Biologist with the U.S. Environmental Protection Agency's National Health and Environmental Effects Research Laboratory. He is the EPA lead for an international effort to develop an Adverse Outcome Pathway, or AOP, knowledge base, which is designed to house descriptions of the biological mechanisms underlying chemical toxicity in a structured manner. Steve serves as a senior advisor in the Office of Research and Development on issues regarding the development of AOP-based predictive toxicology models of disease using genomics, proteomics, and metabolomics. With a combination of experimental and computational experience, he also serves as the liaison with the EPA's National Center for Computational Toxicology and works on web-based tools to support systems biology research within the EPA. Today's talk is titled Accelerating Adverse Outcome Pathway Development via Computationally Predicted AOP Networks. And it'll last about 35 or 40 minutes, and we'll have time for Q&A at the end. As usual, please feel free to type your questions into the chat box, and then we'll hold them till the end of the talk, and then we'll let Steve answer your questions. So thank you, and welcome, Steve.
And thank you, Karen, for letting me speak today, and for everyone online. I guess I'll start with the standard disclaimer that I have to give is I work for the U.S. Environmental Protection Agency, but all the views I express are my own and do not reflect agency policy. And so today, basically, before I get into adverse outcome pathways, I just want to give a brief background on why we care. And the key issue that we have, and let me see if I can get up pointer here. The key issue that we have is that when EPA does regulations, we're dealing with chemicals that are source or chemical contamination in environmental medium. But basically the criteria that we use for those regulations are adverse effects on either humans or wildlife. And so the key issue is how do we connect these two pieces. In addition to that, around the turn of the century, there was a need for, or a recognition of the need for improving the way we do toxicity testing, because the traditional methods are not that predictive, and they are very costly and come at the expense of animal health and welfare. And so there was a concept that was emerged of a toxicity pathway, where instead of looking at these what we call apical endpoints, so in entire organisms, could we do things at a molecular or cellular level that would be predictive of these adverse outcomes? And this we could do, without the use of animals, and also it would be a much higher throughput mechanism for doing toxicity testing that could keep up with the thousands of chemicals that are being introduced, or, well, tens of thousands that are out there, and potentially hundreds being introduced each year. So there were pre-existing some frameworks that addressed the connections between these molecular and cellular events and these apical outcomes. Mode of action was one, but the mode of action tend to be focused on a specific chemical. So the way that it was generated was very chemical specific, and it was designed to be done for one chemical at the time, and that really wasn't scalable for what we needed as far as interpreting results from these high throughput assays that we could do at the molecular cellular level in terms of what they would mean for individuals and populations. And so around 2010, the adverse outcome pathway concept was introduced by Ankley et al. It has, which I will talk about, has progressed through the years, and then another seminal paper on AOPs came out by Villeneuve et al. In 2014. And so what I'll be talking about today is some of our efforts on trying to refine this adverse outcome pathway concept, capture information in a structured form so that it's usable for this purpose of connecting toxicity pathways with adverse outcomes. So the AOP can basically be distilled down into two main components. There are the key events, which are the nodes in the pathway, and key event relationships, which are the edges. The key events are things that represent a change in biological state. They need to be measurable and essential for progression. And we have two specialized key events. So there's a molecular initiating event, which is on the far left-hand side. And this is the initial interaction of a chemical with the biological system. So binding to a receptor or alkylation of DNA or whatever that initial molecular event is, that would be the first step in our pathway. And then on the other end, which is either at an organism or population level, depending on if you're doing a human health or an ecological risk assessment, would be our adverse outcome. And the definition here is it's something that we would care about from a regulatory perspective. So impaired development or impaired reproduction would be examples. Now, again, the key events are really designed to measure and interrogate the system, so we know whether or not an adverse outcome pathway has been activated. But all of the information on how a change in one key event leads to a change in another key event is embedded in these key event relationships. And so, in fact, when I talk about the way we're storing this information, the evidence supporting the AOP ends up getting stored primarily in these edges. And the other thing, which I alluded to in the last slide, is unlike mode of action, these AOPs are chemical agnostic. And the reason for that is if we take 1,000 chemicals and run them through an in vitro screen and we get 100 hits, we want this AOP to be relevant for all 100 chemicals that were positive in that assay, not specifically designed to one. In 2012, the Organization for Economic Cooperation and Development started an AOP development program to both promote international harmonization of how we define AOPs, but also to promote the development and use of this framework. And this was done under the auspices of the Extended Advisory Group for Molecular Screening and Toxicogenomics. This is also the international body that has been overseeing the toxicity pathway-based or high-throughput toxicity testing initiatives. So this represented the logical next step of determining how we can then better interpret those data. This program has resulted in guidance documents, a user handbook, which goes into great detail on how to define these AOPs, and we've also had quite a few training sessions lasting anywhere from half a day to up to about a 2.5-day training with hands-on case studies. We have also, which I'll talk more about in a moment, established an international knowledge base to capture the information. So pretty much all AOPs that are intended to be part of this OECD development program have to go in this international knowledge base. And the advantage to that is we can collect all the information in one place and have that then available to people. And as you'll hear, we're doing quite a bit in terms of trying to capture that information in a way that makes it useful from a computational modeling perspective as well. So the knowledge base is really built up of several modules. So the first module that was released back in 2014 is something called the AOP-Wiki. This module is really tightly tied to the OECD review process. So the AOP handbook describes how to enter data into this wiki module. There's another module called the AOP Explorer, which tends to look at things in a network view, and that'll become clearer as I go through the next slides. We have a third module which deals with more of the quantitative aspects. So this is the module that starts transitioning from a qualitative description of the AOP to something that could actually be used directly to facilitate computational models for that AOP. And then there's an intermediate effects database, which is really the conduit for submitting AOP-related information for regulatory use in Europe. And then we're developing a AOP-KB hub, which will integrate all of these different modules together and provide a single interface for users to go when they want to interact. And while the initial emphasis is on the interoperability among the 4 core modules of the knowledge base, the plan is to make the APIs available for third-party applications and plugins as well. So down the road, we would anticipate that anyone could connect into this system. And the KB is really designed around the different stages of development of AOPs. So the AOP Explorer has a lot of tools for evaluating the existing biological knowledge, and a lot of the work I'm talking about at the end of this talk will feed into this module of the knowledge base. And then basically coming out of that, the idea is you would have one of these things sketched out. So you would have the key events estimated and the relationships between them, but not necessarily have gone through a step-by-step evaluation of that AOP. And that process is facilitated by the AOP-Wiki, where you go through each key event and key event relationship and answer a series of questions to determine the scientific evidence supporting that piece of the AOP. And then, as I mentioned before, Effectopedia captures things in yet a higher, more structured manner to facilitate the computational modeling. And this just shows an example of how the information is stored in the AOP-Wiki. And again, this is tied to the OECD handbook, which is the document that states how you need to describe the AOP if you want the OECD to review and potentially endorse it. And so it's really, the handbook is laid out, describes this as a document, and it actually ends up as a PDF document, which is the thing that gets reviewed by the OECD. But internal to the wiki, we store it not with both an AOP entity, but actually we break out these individual components as separate entities within the wiki. So we can assemble all of this back into a readable document for humans to read, interact with, and review. But, and this just shows, but I'll show in a moment how this storage mechanism allows us to do so much more. But first, let me just show you what one of these looks like. So this is an AOP that is currently under OECD review and is expected to be endorsed this year. It's looking at alkylation of DNA leading to heritable mutations. And this is the diagram of the AOP that the author provided. So we start with our molecular initiating event, which was the alkylation of the DNA. That leads to overwhelming of the DNA repair mechanisms, which then leads to mutations. As these things occur in pre-meiotic male germ cells, then those can be passed to the embryo at conception and then are then inherited in the offspring. The way they would enter this information into the wiki itself is through a table. So you get the same information just captured in a tabular form. You can see over here where we have evaluations of our weight of evidence supporting the AOP, as well as our quantitative understanding of these relationships. And then what we can do is from this tabular view, we can create a network view that shows that shows the AOP. And so you can see it's very similar to the AOP that the authors have drawn. You see these indirect relationships. These are in there because we are very explicit about how the evidence is captured. So if you have evidence connecting your MIE to a downstream key event, we have to have an explicit relationship to capture that evidence. And again, that has benefits down the road. So again, going back to this idea of we store these entities in the wiki, not as a single AOP document, but as a series of connected objects. And the advantage of that is, so now we can enter these things as simple linear pathways, which has advantages from an AOP development standpoint in that it bounds the scope of the problem. But it has disadvantages from a computational modeling standpoint in that all of these things really, in truth, interact. So we've entered these three different AOPs, but in fact they share quite a few key events and key event relationships. And so the way it works in the wiki is once you've defined these entities once, you can reuse them easily. And so not only does this save the author the time and energy of having to replicate the description of these entities more than once, but it means that inside the wiki, these three AOPs are actually stored as a single AOP network. So we have androgen receptor agonism, which through a variety of feedback mechanisms can lead to reduced estrogen synthesis. And what we find is also that inhibition of the steroidogenesis enzymes also lead to the reduced estrogen synthesis. And as the thing progressed, all of these things funnel in on reduced signaling through the estrogen receptor, which could also be due to direct antagonism of that receptor. So now we've captured our three AOPs in terms of one single interconnected network. And this just shows how that is represented in the wiki. So this is looking at it from the standpoint of this aromatase inhibition. So this is the enzyme that leads to estrogen synthesis. And so again, the information is collected in this tabular form, and this shows the network. And what you can see in this network is that there's a series of triangles here. Each one of these represents another AOP that's already in the wiki that overlaps, that contains common key events with this aromatase inhibition. And of the three that I showed in the previous slide, the two outside of aromatase were this one and this one. And so you can see that already we have a more complex network stored in the wiki than even the one I showed on the previous slide. And so you can imagine that over time, if we just allow people to continue to put these things in one pathway at the time, that we'll continue to build up these interconnected networks. And eventually we'll get to something that more accurately represents the biological system, which we understand is much more complex than our current day understanding. And so this process, given enough time, we think would work, but the enough time part is the key point, because you know, building these things up one pathway at the time is going to take a really long time. However, it has been quite productive. So currently in the wiki we have over 100 putative AOPs, though most of these are not under active development at the moment. There are 18 formal AOPs that are currently undergoing some level of OECD review. And the quantitative AOPs are much more difficult. They require a lot more data and time to do, so there's much fewer of those at the moment. However, as I alluded to on the previous slide, we have a need to accelerate this process. And just as one example of why, in the EPA ToxCast program, which I'll talk about in a little more detail in a moment, we have over 400 biological targets that we assay in that battery of high-throughput assays. And we only have approximately 10% of those currently have AOPs in the wiki. In addition, the ToxCast, and even if you include all of the high-throughput testing programs around the world, we still don't believe that we have all of the assays needed to comprehensively cover the different toxicity pathways that would lead to adverse outcomes. And what we'd like to do is get to a point where we're generating the AOPs first and having them guide the assay development. So what my group has been doing over the past few years is looking at data mining methods that we could use to generate what we're calling computationally predicted AOPs that can then feed this expert-driven process. And so basically the concept here is we're reversing the process that I showed in the previous slide. So instead of having a true network that's built up over time, we're going to use network inference algorithms to come up with an estimate of what this true network would be. And then we're going to use network traversal algorithms to extract subnetworks from this larger network that are amenable for human evaluation. And then what we'd like to do is feed that into the expert-driven process. We have two papers. One is out, one is to be out soon, describing this process, the making of the global networks. And then we have one paper that's in preparation where we look at extracting these smaller subnets from the larger global net. And this just shows the overall process. So we're, at this point, relying on public data sources, and I'll describe that in more detail coming up. We generate these estimates of our interconnected global network, extract these computationally predicted AOP subnets, feed those into the expert review process. Here they get corrected and amended, and then the correct version gets entered into our knowledge base. So that contributes to our true network, our true global network that's being formed down here. But it also feeds back up here because we get information on of the predictions we made, which edges were correct, which edges were incorrect, and which edges did we miss. And so then we should be able to use that to refine our methodology for both the global network construction as well as the subnet extraction. There are many data sources. This just shows a few examples. The main point to make here is that what we're looking for are data sources that cross as many levels of biological organization as we can get, because again, the key here is we want to be able to map these things that we're measuring in high throughput to adverse outcomes that would be over on this end. The 4 data sources I'll talk about today are the ToxCast data from the EPA, T.G. Gates dataset, which is a large toxicogenomics dataset from Japan, the comparative toxicogenomics database, and we use the Reactome molecular pathway database for a lot of our analysis. The first case study I'll describe is using ToxCast and the comparative toxicogenomics database. So the EPA ToxCast program is basically a high-throughput screening program for toxicity testing. Comparative toxicogenomics database is hosted by NC State, and it contains chemical to gene associations as well as chemical to disease associations. The ToxCast program currently has over 1,800 chemicals that have been screened in at least some of the assays, has over 700 assays that monitor over 400 biological targets. And it's also part of a broader effort within the U.S. Called the Tox21 effort, where in a certain number of assays, we've actually screened 10,000 chemicals in a smaller number of assays. But the ToxCast assays were more amenable for our work because we have a much higher number of assays. The comparative toxicogenomics database has done literature curation to determine both chemical to gene and chemical to disease associations. So the gene associations come both from genetic databases such as OMIM, but also from published toxicogenomic studies where they found changes in gene expression in response to certain chemicals. The disease associations come both from literature-curated information as well as inferences from the toxicogenomics data. So from these two data sets, we're going to use a method called frequent item set mining, and this is what online retailers will use often to define what you might be interested in based on past purchases. There's many references could be given. This particular reference includes the description of the R package that we used, which is the A-Rules R package. And I'll go through the meanings of these different terms over the next few slides. And at the end of the day, all of this can be derived from the contingency table when you look at these different transactions. So the support for either a rule, which is any pair of items, or for the items themselves, is simply the probability of occurrence of that pair or that item in the data set. The confidence is the conditional probability of seeing one of the items. So in this case, it's showing milk given the other item, eggs. And again, this can be derived from the contingency table. And then the lift is actually comparing the probability or the observed association between any two items and comparing that to the expected number of associations given the assumption of independence. And once again, this can be derived from the values in the contingency table. And I'm guessing this audience is well familiar with these techniques. And so what we've done for our purposes is we've adapted this for the chemical data. So rather than using a transaction from a store, we're starting from a given chemical, and we're looking at these different toxicity measures. So whether a chemical had activity in a high-throughput assay, whether the chemical affected the gene expression of a gene, or whether the chemical has been previously associated with a disease. And we just use these as our items in our transaction. And so, and we can do this because of this fact that the AOPs are chemical agnostic. And so what this allows us to do is to define the AOPs in terms of a broad number of chemicals rather than specifically looking at the AOP for one chemical at the time. So when we do this, so we basically take our ToxCast data and we discretize it. So now that we're saying basically, was the chemical a hit in that assay or not? Our CTD data is already discretized, so that comes as associations between the chemical and either the disease or the gene expression. We can now use the frequent item set mining. In our case, we compared the ToxCast information with the CTD disease, and then we compared the CTD disease associations with the gene expression from CTD. And so what we get is a series of associations, and what we've been using as our edge weights for these associations are these lift scores that we would calculate by comparing the observed number of co-occurrences to the expected number of co-occurrences with the assumption of independence. And so then to determine whether or not these networks are meaningful, we then query these networks using either adverse outcomes of interest or, in the case I'll show you, we started from a ToxCast assay of interest, and then look at the neighboring nodes to see if they replicate anything that would be expected from the literature. And so this is just showing one example where we looked at the aryl hydrocarbon receptor, which is one of our assays in ToxCast. And we said, okay, show me the connected diseases, the associated diseases, based on both the ToxCast data as well as the CTD gene expression data. And what we were struck by is that in ToxCast, and so the way to read these figures is the AHR is in the middle, and the distance from the AHR is inversely proportional to the association. So things that are towards the center of this circle are highly associated with the AHR. Things out to the periphery are less associated with the AHR. And so what we found is that glaucoma, there were several disease endpoints related to glaucoma, and they were very prevalent in the top hits for AHR in ToxCast. while those same associations were seen in CTD, they weren't among the strongest hits. They were a bit weaker. So to explore this further, we then took those glaucoma disease endpoints, which are now shown here in blue, and we queried the same network and said, okay, show us the the genes now that are most associated with the diseases. And there's a number of genes, and you could talk about this for a long time, but what I would like to highlight for this talk is that we, as you would expect, got the AHR gene from our ToxCast data set. We also saw the AHR in our CTD data set, but we also found this other gene, which is CYP1B1. And with some follow-up work looking into the literature, we were able to determine that, in fact, there's quite a bit of literature looking at AHR and glaucoma, and this CYP1B1 is regulated transcriptionally by the AHR receptor and is predicted to be the mechanism by which AHR influences glaucoma. And there's been genetic polymorphisms in this gene which are definitively associated with the glaucoma disease. This also served as a great technical example for how these things work because on the ToxCast side, we picked up AHR. Again, this was our strongest association. This is what started the whole thing. But CYP1B1, even though it is in the ToxCast battery, was not picked up. However, that makes sense because the ToxCast assay is looking for direct inhibition of CYP1B1. That's not what these chemicals do. These chemicals are changing the levels of CYP1B1 in the cell. In CTD, we picked up AHR through our methods, but if you were to do a query for CTD, you would not pick up the AHR to glaucoma connection. That's because CTD tends to rely on gene expression data, and these chemicals do not change the gene expression of AHR, at least not dramatically. They directly interact with AHR to change transcription of genes such as CYP1B1, which is why we picked that up in CTD. So we thought this was not only a great example that, yes, indeed, these global networks we're generating seem to have real biology, but it also showed the value of incorporating multiple data sets into this analysis. We have a second case example where we started with a raw toxicogenomics data set. This is out of Japan. It was a large study looking at 170 chemicals in dose response and time course. They did a variety of studies. We focused on the in vivo liver endpoints, and what that gave us was toxicogenomics data as well as phenotype data. And the phenotype data was in the form of liver histopathology as well as clinical chemistry. And the omics data, we were able to do pathway enrichment, and so this gave us hit calls for is this pathway enriched or not in response to a given chemical. We can compare that to calls on the phenotypes, whether the histopathology call was positive or negative, or whether the clinical chemistry was out of the normal range or not. And so in doing that, we can use the same frequent item set mining, generate this global network. We can do queries from that global network. In this case, our queries were focused on a disease, which was fatty liver. And then we can go through and manually annotate at this point the pathways and the phenotypes that come out and try and group them logically. And then you can use that with some expert judgment to start assembling these putative AOPs. So to make sure we were on the right track, we took carbon tetrachloride, which was a chemical in the TG Gates data itself, and we said, okay, now we've got this network, let's look at what perturbations in that network are driven by carbon tetrachloride and compare that to the known mode of action. And what we found is that of the key events we identified independent of the previous literature, we had very good coverage of the overall mode of action. There were a couple of other cases where once we had the mode of action, we could look in our data and find support for it, but we did not come up with these things independently. And then there are a few cases where we just missed key events, and most of these are cases where the data we have thus far would not inform us. So we do not have enzyme activity outside of the ToxCast data, which were not used in this study, and we don't have metabolomics data to get things like lipid peroxidation. So again, these are things that should be overcome by incorporating additional data sets. So then, like I said, the ultimate goal is we want to be able to leverage as much data as we can. So the next step in this process is to be able to take multiple networks, merge those networks, and start extracting these cpAOP subnetworks. Key things that need to happen for this, we need to map to a common vocabulary, standardize our edge weights, identify network traversal algorithms that can identify these communities, and then extract those communities that would be relevant for a given adverse outcome. Just to quickly talk about ontologies, so we have developed a very minimal AOP ontology that captures these key components, or our key events, our key event relationships, and connecting those. We have entities that connect those to an AOP. And what we're in the process of doing now is trying to define these events in terms of additional biological ontologies, such as the gene ontology or cell ontology or Uberon. And these ontologies are tied to these levels of biological organization that go into our AOP. We also need to standardize the lift across the data set. So this just shows an excerpt of the results that we get using the ToxCast data in that first case study that I showed versus the CTD data. And you can see that the numbers just in general are inflated in the CTD data relative to the ToxCast data. And this is due to the fact that the CTD data had much more data in it. And so these are real inflations, but they cause issues when we're trying to now merge these networks. So to standardize the lift across the data sets, we can take advantage of a publication by McNicholas et al., where they actually calculated what the theoretical minimum and maximum values could be for a lift given a specific rule. And so this lambda value represents the minimum value that you could get for a lift. This V represents the maximum value you could get for the lift. And so now if we take our lift score, subtract out this minimum value, take our max, subtract out this minimum value, now we have everything tied to 0. And if we now take the ratio of our lift to this max value with both of them, with them tied to 0, we can get a proportional score that goes from 0 to 1, and this is going to be consistent across the different data sets. For community detection at the moment, we do a random walk algorithm, which is implemented in R as cluster walk trap. And so the goal here is, if you recall in the manual example I showed earlier, I said we manually pulled together groups of pathways and phenotypes and then labeled those as our key events. So we're trying to automate this process, and this is the first step in that process. And then once we have our key events that we've estimated based on these communities that we've generated, then we can query, as we've done before, query with an adverse outcome or a target of interest. And this just shows an example. So this is fatty liver disease, as I showed you in the previous example, except in this case we've queried it having previously done the community finding to generate the key events. And so now, whereas before those groups of pathways and phenotypes were all done manually, now these things have been done automatically. This just shows the different pathways and phenotypes and the community into which they were assigned. And we can actually, in this case, since we had the TG Gates data, which has time course information, we could actually take these key events and order them, and we could provide, based on the pathways and phenotypes that were in a given key event, we could put labels on these. So at the moment, these things were manually done. because the ontology work is running in parallel to this process. But again, the long-term goal is once we have these things tied into that ontology work, these labels even should fall out of the automated process. And of course, our eventual goal is to feed this information into the expert-derived process. So this just shows, again, focusing on fatty liver, shows some expert-derived putative AOPs from a recent publication by Michelle Angrish et al. And even though our methods were running in parallel to her generating these putative AOPs, she's actually been involved in evaluating our work and has agreed that, you know, if we had been upstream of her effort, that the information we had would have been valuable in this process. So hopefully I've convinced you that what we're able to do is that we are able to generate, you know, some approximation of these global networks, that we're able to extract some sub-networks from these larger networks that are also meaningful, and that there is a process in place between what we're doing on the computational side and the tools we put in place on the AOP knowledge base to start getting a virtuous cycle whereby the expert reviews improve our ability to make automated predictions, which then accelerate the expert reviews. And just to acknowledge everyone, so on the left, this work is highly collaborative, and to put everyone from all of these teams on a slide would take too much. But there's a large team within the EPA that does AOP discovery and development. Highlight a couple of folks who were particularly helpful for these slides. We have an international working group defining the knowledge base. There's the extended advisory group that I mentioned. We've worked very closely with the comparative toxicogenomics database team, especially Carolyn Mattingly, who runs that project. The wiki development was done primarily by a bunch of really talented student contractors at the EPA. And then the cpAOP work that I focused on at the end, most of that work is Nofasat Oki, who's a postdoc in my group, and Shannon Bell, who's a former postdoc in my group, with help from Kataya Eyes and Chris Grant. And I missed you somehow. Mark Nelms should also be on this list. Yep, and I think that's it. So I can take questions, or if we have questions.
Yeah, so you guys can feel free to type any questions you have in the chat box, or you can unmute and just speak up.
And I should say while they're typing that, you know, one of my goals in here, so obviously we have been pushing to get something that works, but one of my goals in giving this talk is that we don't claim to be experts in all of these techniques. And so, you know, really I was posing this as much as a problem for anyone interested in solving it as a problem that we have come up with at least one solution for. So, so certainly if anyone's interested in collaborating on methods for any of these steps, you know, we'd be very interested in talking with you.
I don't see anything coming up in chat.
Well, while people are talking and getting their question together, let me ask. It looks like y'all are making heavy use of curated structured database. Have you started looking at automated extraction of literature?
Looking, yes. That's really not an area we want to go into. It's certainly an area we'd love to tap. And so absolutely, if there's anybody who's kind of doing that and could give us the outputs from that, then we would love to have those as inputs to our work. But yeah, that would be a logical extension to this is if we could get to the point where we can pull information directly from the literature.
Okay. All right. I don't know. We're not to the results stage yet. Don't have anything you can just click on and pull into R right now, but
yeah.
Okay. All right.
Oh, sorry, and there was a question to repeat the question. So the question was on if we had, so what we've been focused on, and even the data sources that I highlighted up there were curated databases. So a human has been through the literature and has made calls on, these associations are real or not. Or in the case of toxicogenomics, we were directly using, raw data directly. And the question was, have we looked into any type of text mining that would give us a much broader swath of information? And my answer, which you heard, was we're very interested in that because it would help. One of the issues we have right now is there is a lot of bias in these curated databases towards certain adverse outcomes that have been highly studied. And obviously we want to cover all of biological space. So hopefully the text mining from the literature would help with that. But again, our focus has really been on this problem, and we just don't have the bandwidth and really not the expertise to get into the text mining. So if anyone is out there, including Eric, who gets some results and would be interested in working with us, we would love to do that.
Okay, so I'm going to start closing things down, and if anybody comes up with any more questions, you feel free to type them in while I'm doing that. But I want to thank you, Steve, for presenting your work today. We really appreciate your time. And for those of you who are logged on, and we just have a few NCDS reminders, I want to make sure you guys know about Data Matters, which is a week-long series of 1- and 2-day courses aimed at data professionals, which you guys probably are. Registration is open, and early bird is going until May 15th. So if you're interested in that, you can go to datamatters.org to check it out. Also, we do this DataBytes thing every month, and our next session is going to be Wednesday, June 1st at noon. It's going to feature Tony Hay of the United Kingdom Science and Technology Facilities Council. So hope to see you then. And I think that closes us out. So thank you.
Thank you.
All right. Everybody have a good day.